Guys, good morning. You are welcome to my YouTube channel, the Explicit Tutors. My name is Tutor Joseph. In today's video, we shall be looking at compiled exam-likely questions on Tutor Monera by Tutor Joseph. I did the compilation myself. Before I get started, excuse me, please. Let me give us a piece of advice. Now, should I say you now? We. Okay, let me include myself. We just gain admission into the University of Benin, and you would be told to read certain courses. Some are easy, some are difficult to assimilate. Okay, I must be realistic here. I can't tell you that all is well. No, all is well, definitely, but it is good you say the facts. Some courses are really difficult to understand. That's why tutorials are there to help you with those difficult ones. Now, one of these difficult courses is PBB 111, which is what? Plant biology and biotechnology. And the first topic that you guys are going to read is living things, like general characteristics of living things. Of course, I'm not taking it, it's quite easy. Then number 2, you have the microscopy, how microscopes are used. And chapter 3 is what? Kingdom Monera, and that is the one I'm taking right now. I've taught it as a topic already. Everything about it has been taught. Now what we have to do right now is we have to revise questions, that is, we have to tackle exam likely questions, so that on the day of the exam you won't develop cold feet. Please, as you are watching this video, endeavor to subscribe, like, share and comment. Let us get started. Please, before we start, get a chill water or cold mineral, so that as you are going through the questions, they are entering into your cerebrum. Number 1 says, which of these was formerly called blue-green algae? Actinomycetes, cyanobacterium, chlorobacteria and the streptococci. The actinomycetes weren't called that, the streptococci weren't, as well as the chlorobacteria. So the answer is what? Cyanobacterium. Number 2 says, the third group of bacteria, which was proposed in year 1976 by Ralph A. Lewin, possessing chlorophyll A and B, phycobilins and accessory carotenoid pigment, were A. Actinomycetes, B. Cyanobacterium, C. Chlorobacteria, D. Streptococci. The answer is the chlorobacteria. The chlorobacteria possess the aforementioned characteristics. That is the characteristics mentioned in number 2. Number 3 says, which of these contains nitrogenase? Could it be the actinides or the heterocysts, the heterocytes, and the mucilage? Now looking at question option B and C, they are very close. Okay, and one of them is the answer. Is it the heterocyte, which is C, or the heterocyst, which is B? Please, the answer is B, heterocyst. Watch my videos and you get to know these things. Number 4, vegetative cells that enhance reproduction and possess enzyme nitrogenase is the dash. A. Akinete, B. Heterocyst, C. Heterocyte, and D. Mucilage. What is the answer that possesses vegetative cells and also possess nitrogenase? So the answer is heterocyst, B. Okay. Number 5, dash reserves food for adverse condition. The answer is the akinete. The akinete does that. Now let's look at question number 6. The 2 filamentous cyanobacteria are A. nostoc and anabena, B. oscillatoria and cleocarpus, C. pandorina and volvox, D. ulothrix and oscillatoria. Please, the 2 filamentous cyanobacteria are the nostoc species and the anabena species. Both of them are called filamentous bacteria. Number 7, which of these is not correct about nostoc? Its heterocysts are both intercalary and apical. B. It is both terrestrial and aquatic. C. Its filaments form tangled, twisted colonial cells. D. It has a more occurring akinete. The answer is D. The nostoc species doesn't have a more occurring akinete. Rather, the anabena has a more occurring akinete. It therefore means that the nostoc species has less occurring what akinete. So the answer is what? D. Number 8. The smaller filaments formed via fragmentation are called heterogonia, B. macrogonia, C. homogonia, and D. homogonia. The answer is C. homogonia. Please take note of that. Don't get confused by these options. Okay, just go straight to the answer. The answer is what? D. homogonia. Number 9. Dash produces through homogonia. Good. A. We have Oslotera princeps. B. We have Glucarpus quartnata. C. We have Ulothrix zonata. And D. We have Pandorina morum. So the answer is Oscillatoria prince. Okay, through homogonia. Please, the answer is Glucarpus quartnata. B. The answer is Glucarpus quartnata. 10. Which of these undergo normal fragmentation with akinete? You know, when I'm looking at question 7, I made mention of akinete, or the nostoc species and the anabena have akinete. They undergo reproduction, that is normal fragmentation, which is a type of asexual reproduction using the word the akinete. Please take note of that. So now let's go to number 10. The ministry is moving. I believe all these things they are entering. Just watch my video first before you attempt these questions, okay? Number 10. Let's take number 11, please. Which of these have the ability to fix both nitrogen and release oxygen? A. Could it be the actinomycetes, or the cyanobacterium, or the closed bacteria, or the streptococci? Please, the cyanobacterium has the ability to fix both nitrogen and release oxygen to the atmosphere because it is what? Photosynthetic in nature. It uses oxygen. It uses water. You know, it's from water molecule that oxygen is liberated. Please. Number 12 says, which of these is wrong about bacteria? All right, let's see. Seen using the electron microscope, I don't know yet. B. Possession of plasmid. C. Seen using the light microscope. D. Absorption through cell wall. Please take note of this. The bacteria are not seen using the electron microscope. So the answer is what? A. They are seen using what? The light microscope. You can only use the electron microscope to view what? Viruses. Number 13. Which of these is odd? A. Methanogens. B. Thermoacidophils. C. Halobacteria. D. Cyanobacteria. The odd one here is the cyanobacteria. That is because the kingdom Monera has two subdivisions. The kingdom Monera has two subdivisions. We have division 1, Archaeobacteriobionta. Then 2, we have Bacteriobionta. All these are the two subkingdoms, or two subdivisions. Okay, it's subkingdom rather. Two subkingdoms of kingdom Monera. Subkingdom means the kingdoms under the actual kingdom. Okay, please, the methanogens, which are the methane bacteria, they are found in swampy, marshy places. They are also found in sediments and the intestinal tract of what? Animals. Okay, so they have the ability to produce what? Methane. You know, methane is called what? Marsh gas, and it is the principal constituent of cooking gas. It is very, very what? Volatile. Then B. Thermoacidophils. They have the ability to survive in sulfonic and acidic region. And C. Halobacteria. They best survive in salty environments. So the odd one here is what? The cyanobacteria, which is what? Option D. Number 14, let's take it. Which of these is found living in acidic and sulfur environments? I think I've already explained that. The answer is what? Thermoacidophils. Thermoacidophils. Number 15, dash are found in swampy, marshy places, sediment and intestinal tract of animals. I did explain it just now. The answer is the methanogens or the methane bacteria. Now that is just the answer. Very, very easy. Just read. All these things will be piece of cakes, okay? Let's take number 16. Which of these looks like chloroplast in green plants? Could it be the pleuchloron, B, the pleuchloron-3, C, the metachloron, D, mesocloron? So long Monera is concerned, mesocloron and metachloron do not exist. Metachloron do not exist. I just coined the both of them. So the answer is between A and B. The answer is actually the pleuchloron, which is what? A. Number 17. The father of bacteriology is A. Louis Pasteur, B. Robert Koch, that is Robert Koch, C. Robert Brown, D. Anthony von Leeuwenhoek. So the answer is the father of bacteriology. The answer is Louis Pasteur. Louis Pasteur was the first to discover bacteriology, or to put down the meaning of what? Bacteriology, which is the study of what? Bacteria. Number 18, Dash was the first to discover bacteria. Please, Anthony von Leeuwenhoek was the first to discover the bacteria. Okay, take note of that. Number 19, the rules to diseases caused by microorganisms were first stated by Robert Koch. Robert Koch was the first to put down anything that has to do with diseases, okay, caused by microorganisms. Please, Robert Koch was also the first scientist to discover the causative agent of tuberculosis, and the causes or the causative agent of tuberculosis is the Mycobacterium tuberculosis. Take note of that. And he did this in year 1822. That was the year he discovered Mycobacterium tuberculosis. Number 20. Which of these are STDs? A. Syphilis and gonorrhea, B. Leprosy and tuberculosis, C. Gonorrhea and measles, D. Syphilis and tuberculosis. The answer is syphilis and what? Gonorrhea. Syphilis is caused by a bacterium called Treponema pallidum. Then gonorrhea is caused by a bacterium called Neisseria gonorrhea. And all of these bacteria are negative gram bacteria. Please, the answer is what? A. Number 21. Syphilis is caused by which of these? Okay, it's even here. The answer is what? Treponema pallidum. Oh, this option A, pallidum, was wrongly spelled. It ought to have been P-A-L-L-I and not A, okay? So the answer is what? A. Let's take number 22. I believe the ministry is going on. Please take some water. I would love to even drink some water right now. Number 22. Which of these causes typhus fever? I never said typhoid fever. Typhus. A. Treponema pallidum, B. Rickettsia prowazekii, C. Chlamydia psittaci, and D. Mycobacterium species. Please, Rickettsia prowazekii causes typhus fever. Causes what? Typhus fever. Typhoid fever is caused by Salmonella typhi. Typhoid fever is water and foodborne disease. Typhus is caused by Rickettsia prowazekii, but typhoid fever is caused by Salmonella typhi. Please take note of that. Number 23, a disease that leads to blindness called trachonema is caused by Chlamydia psittaci. Please take note of that, which is option C. Chlamydia psittaci causes what? Trachoma, which can lead to what? Blindness. Number 24 says, which of these is responsible for the massive growth of tree trunks? A. Pseudomonas aeruginosa, B. Pseudomonas putida, C. Acrobacterium tumefaciens, and D. Bacillium temulivorans. All right, the answer is what? Acrobacterium tumefaciens. Agrobacterium tumefaciens are responsible for the massive growth of tree trunks. Take note of that. Number 25. Dash degrades trichloroethylene. Dash degrades trichloroethylene. The answer, okay, option A says Pseudomonas aeruginosa, B. Pseudomonas putida, C. Agrobacterium tumefaciens, D. Bacillium temulivorans. The answer is Pseudomonas putida. Yeah, B. Pseudomonas putida causes or it degrades trichloroethylene, okay, or trichloroethene, as the case may be. Number 26. Dash degrades naphthalene. Please, Pseudomonas aeruginosa degrades normal hexadecane. So the answer is Bacillium temulivorans. Bacillium temulivorans degrades naphthalene. Take note of that. Number 27 says dash degrades N-hexadecane. Is this N-hexadecane, right? N-hexadecane. So the answer is temulivorans. Okay, the answer is we have option A, Pseudomonas aeruginosa. Excuse me. We now have B, Pseudomonas putida, and we have C as Agrobacterium tumefaciens, and we have D as Bacillium temulivorans. So please, N-hexadecane are degraded by Pseudomonas aeruginosa, that is Pseudomonas aeruginosa, which is what? Option A. All right, the next one says which of these is not used as bioremediation of toxic waste? Pseudomonas aeruginosa, Pseudomonas putida, Agrobacterium tumefaciens, Bacillium temulivorans. The answer is what? Agrobacterium tumefaciens, and it is actually what? Parasitic in nature. Please take note of that. It is very, very important. Now number 9 says which of these is not a beneficial bacteria? Which of these is not a beneficial what? Bacteria. Okay, we have Rhizobium japonicum, we have Clostridium pastorianum, we have Azotobacter indicum, and we have Agrobacterium tumefaciens. Of course, the answer is what? Agrobacterium tumefaciens. They are not beneficial, they are what? Pathogenic. That is, they are capable of causing diseases. Number 30 says mold-like bacteria are called A. Actinomycetes, B. Cyanobacteria, C. Chlorobacteria, D. Streptococci. Please, the answer is Actinomycetes. Now, let me quickly drop a note on this part. Actinomycetes israeli causes oral cavity disease. That is the disease of what? The mouth is caused by what? Actinomycetes what? Israeli. Then Streptomyces venezuelae produces an antibiotic called what? Chloramphenicol, and it is used against intestinal tract disease. You also have a Streptomyces erythromycin. The Streptomyces erythromycin, they produce an antibiotic called erythromycin, which is used against what? Whooping cough. Whooping cough. You now also have streptomyces. We have one again. Okay, yes, streptomyces griseus. Streptomyces griseus produces streptomycin, which is used against what? Bacterial infection. Okay, so please take note of that. I said it because I don't know if I compiled questions on that aspect. Number 31 says, which of these is not a nitrifying bacteria? A. Rhizobium japonicum. B. Clostridium pastorianum. C. Azotobacter indicum. D. Clostridium denitrificans. As the name implies, denitrification or denitrification. The answer is what? D. Take note of that. Number 32, the ministry is moved. We are almost running off. Dash is made up of prochloron and prochlorothrix. The answer is chlorobacteria. Please, it is not the actinomycetes, it is not the cyanobacterium, and it is not the streptococci. It is what? The chlorobacteria. Number 33, which of these are gram? Okay, all of these are gram negative bacteria except A. Mycobacterium tuberculosis, B. Treponema pallidum, C. Neisseria gonorrhea, D. Escherichia coli. The answer is Mycobacterium tuberculosis. Yes, Mycobacterium tuberculosis is positive gram bacterium. Okay, number 34 says all are gram positive bacteria except A. Mycobacterium tuberculosis, B. Clostridium titani, C. Staphylococcus aureus, D. Cyanobacteria. Please, cyanobacteria is a negative gram bacteria. That becomes the answer. D is the answer. Number 35 says, which of these were formerly classified as viruses because they could occur as intracellular parasite of eukaryotic cells? A. Rickettsias and Spirochaetes. B. Chlamydias and Spirochaetes. C. Rickettsias and Chlamydias. D. Cyanobacteria and Spirochaetes. So the answer is Rickettsias and Chlamydias. The answer is what? C. They were formerly called viruses. Please, now these organisms, they have peculiar characteristics that distinguish them from every other monerans. Normally, viruses possess DNA or RNA and never the both. Viruses possess DNA or RNA and never the both. But these guys, the Rickettsias and Chlamydias, they possess DNA and RNA. They also have ribosomes. And they also have enzymes that help them to produce their own nucleic acid. You can see that they are very, very different. Okay, they have enzymes that give them the ability to produce their own nucleic acid. So they are distinguished types of viruses. Please, the answer is Rickettsias and Chlamydias. Number 36 says the protein coat surrounding the virion is called A. Plasmids, B. Capsule, C. Capsid, and D. Leptoid. So the answer is what? Capsid. The capsid is that protein coat that is found surrounding the virion, which is what? The non-living coat of bacteria. Please take note of that. That is outside the living cell. Outside a living cell, the bacteria. Outside the living cell, the viruses are known to be non-living, called virion, which means that virion is a non-living virus. Okay, that is number 37 says T-shaped Ebola virus called dash causes hemorrhage. A. Bacteriophage, B. Actobacteriophage, C. Mosaic virus, D. Adenophage. Please, the answer is what? Bacteriophage. Bacteriophage is a T-shaped Ebola virus that causes what? Hemorrhage, internal what? Bleeding. Number 38 says, which of these is false about a virus? A. Possession of DNA or RNA, which is definitely true. B. Possession of capsid, which is definitely true. C. Possession of plasmid. I don't think that is true. And D. Seen with electron microscope. Yes, viruses are seen using what? The electron microscope, but they do not possess plasmid. Bacteria only possess what? Plasmid. Plasmid is the extra chromosomal DNA found in the bacteria, or you can say it is the extra DNA strand found in the what? Bacteria, and it is possessed by what? Bacteria only. Viruses do not have plasmid, they only have capsid. Okay, number 39 says, Eubacteria undergo genetic recognition by A. Conjugation, B. Transformation, C. Transduction, D. All of the above. The answer is what? All of the above. They undergo conjugation, transformation, and what? Transduction. Okay. Number 40 says, during the experiment of Christian Graham in 1884, the mordant used was dash. A. Violet dye, B. Sofranin, C. Iodine, D. Alcohol. Please, the mordant used was iodine. Alright, the mordant used was what? Iodine. Mordant is a substance that has the ability to stick something to what? Itself, like it's gummy. It gums or sticks something to itself. That is what? A mordant. And during his experiment, iodine was used as what? The mordant. Number 42 says, the stain used was dash. A. Violet dye, B. Sofranin, C. Iodine, D. Alcohol. Please, he used a sofranin stain. All right, please take note that. Number 3, which of these was responsible for the color change of from the color change blue to purple of the positive gram bacteria? A. Could it be the violet dye? B. Could it be the stain, which is the sofranin? C. Could it be the mordant, which is the iodine? D. Or the alcohol? The answer is the violet dye. Yes, the violet dye is responsible for the color change, which is blue to purple of what? The positive gram bacteria. Number 44 says, the negative gram bacteria appeared dash, pink to red, B. Blue to green, C. Pink to brown, D. Orange to yellow. Please, they appeared pink to red and not pink to brown. Please, the answer is what? A. Please, pink to what? Red. Gram-negative bacteria appeared pink to red, but gram-positive bacteria appeared blue to purple. You can also say that the gram-positive bacteria are bacteria that have a lot of peptidoglycans. They have a lot of what? Peptidoglycans. While the gram-negative bacteria have little of what? Peptidoglycans. Peptidoglycans are the cell wall of what? Bacteria, and peptidoglycan is a polysaccharide that is what? Interconnected, that is made up of what? Amino acids. Okay, it is made up of what? Interconnected polysaccharides, amino acid combined with what? A muramic what? Acid. Interconnected amino acid and polysaccharides and muramic acid is which of these? They may give you cell wall, peptoglycans, glycocalyx, and other useless option. Please, the answer is what? Peptidoglycan. Let's take question 5. Okay, we have about 5 questions to go. We have about 5 questions to round off this video. Number 45 says, pleuropneumonia-like organisms are also called A. cyanobacteria, B. actinomycetes, C. mycoplasmas, and D. chlorobacteria. The answer is what? Mycoplasmas. The mycoplasmas were formerly called, or are called, PPLO, which is what? Pleuropneumonia-like what? Organism. Mycoplasmas are also called what? Pleuropneumonia-like what? Organisms. PPLO. Number 46, culling of leaves. Stunted growth and yellowing of leaves are caused by A. Rhizobium japonicum, B. Clostridium pastorianum, C. Azotobacter indicum, D. Mycoplasma gallisepticum. Mycoplasma gallisepticum is found in, is the causative agent of culling of leaves, stunted growth, and yellowing of leaves in plants. And it is majorly found in the phloem of plants. It's a very bad guy. Please, it is a very bad guy. Mycoplasma gallisepticum is found in the phloem of plants. You know that the primary function of the phloem is to carry manufactured food through translocation. As soon as food are manufactured, the phloem carries it through translocation to other parts for what? Survival. But because of this satanic agent, Mycoplasma gallisepticum is always causing interruption. Therefore, the food molecules manufactured are not able to be translocated, resulting to culling of leaves, stunted growth, which is slow growth, and yellowing of what? Leaves. Satanic what? Agent. That is not option D. 47. Which of these are known to be the smallest organisms, which are even smaller than the bacteria? Could it be the cyanobacteria? I don't know yet. Could it be the actinomycetes? Could it be the mycoplasmas or the chlorobacteria? Please, the smallest organisms on earth are called mycoplasmas or pleuropneumonia-like what? Organism, which is C. Number 48, the cell wall of eubacteria is called A. peptidoglycan, B. pseudopeptidoglycan, C. cellulose cell wall, D. polypeptidoglycan. I love these options that I coined. I too much. So you choose which of them do you think is the answer, because I explained it not quite long. The answer is what? Peptidoglycan. Please, I told us that the kingdom Monera are divided into sub-kingdoms, which are the Archaeobacteriobionta and the Eubacteriobionta. The Archaeobacteriobionta do not have peptidoglycan, but the Eubacteriobionta, they have peptidoglycan. So which means that eubacteria possess peptidoglycan, while the Archaeobacteriobionta possess pseudopeptidoglycan, which is what? False. This pseudo, pseudo, means false, which means that it possess false peptidoglycan. So at this point, we call it a day. If you know you have learned something, please endeavor to subscribe to my YouTube channel. Please, thanks for watching, and do have a wonderful day. Just go through these videos, go through them over and over again for a better understanding. Please, before you answer these questions, make sure that you must have listened to my video or you must have read this topic. Do not depend on past questions. I repeat, do not depend on past questions. Thanks for watching, and do have a wonderful day.